I'm Senjuti Saha. I'm a microbiologist at the Child Health Research Foundation in Bangladesh. When I was 17 and in Bangladesh, I stole my parents' credit card to apply to schools in Canada. About 12 years after that, and in 2016, with a PhD in molecular genetics, I came back to Bangladesh and teamed up with my father to run a not-for-profit organization called the Child Health Research Foundation, and also to pay back those application fees. Today, our NGO has over 300 members, and we focus on reducing the burden of infectious diseases in children through research and advocacy. When I came here, my mission was to specifically fight meningitis, an infection of the brain that often leads to death or lifelong disability. Turns out that for a majority of meningitis cases, we still do not understand what pathogen exactly causes it, a bacterium, a virus. If we don't know what causes a disease, we can't treat or prevent it. I figured that we could use genomic sequencing to get to the bottom of this mysterious meningitis cases. But we did not have that facility in Bangladesh. Finally, 2 years ago, overcoming many barriers, with the help of amazing collaborators, we set up a small sequencing facility in our lab in Dhaka. Using sequencing, we have been finding pathogens that we never knew causes meningitis, like chikungunya virus. But even more importantly, in the process of setting up a sequencing facility in Bangladesh, we trained local young scientists at the front lines of public health. And turns out, when you empower, engage, and educate young people, unintended consequences follow. While we might have gotten that sequencing machine for meningitis, our team used it to sequence dengue virus in 2019 during one of the worst dengue outbreaks in Bangladesh. Also in 2019, we started sequencing respiratory viruses like influenza and RSV. But really, the importance of building local capacity came to the front when COVID-19 hit. The first 3 cases in Bangladesh were detected on March 8th, but there were not many testing labs at that point. And so our country, like many others, had to quickly ramp up testing. On March 24th, the government of Bangladesh called us to work as an extension of the government testing facility. And it made sense. Our NGO has been conducting surveillance for decades. The data we generated in the past facilitated the introduction of hip and pneumococcal vaccines to fight pneumonia and meningitis in Bangladesh. And as I mentioned earlier, we also had experience in respiratory disease surveillance. So we got to action. We immediately reorganized our labs, got more biosafety cabinets, ordered PPEs, arranged refresher trainings, you name it. We even got accommodations ready so team members did not have to commute during lockdowns and to keep families safe. Our gym, dining room, storage room all became COVID-19 response rooms. Within 5 days, by March 29th, we started testing. We were prepared to work 24 hours in 3 shifts, handling hundreds of samples. However, there was a problem. We were receiving only 10 to 12 samples per day. We discovered 3 main barriers. Barrier 1, lack of knowledge on where and how to give samples. To resolve this issue, we immediately opened up hotlines for anyone to call us and ask their questions so we could try to answer them. Barrier 2, many hospitals or clinics, despite wanting to collect samples and increase the number of tests, lacked the experience of collecting samples safely and sending them to testing labs. We rolled out a Train the Trainer program and trained 16 teams on respiratory sample collection, transport, and proper use of protective equipment. Barrier 3, lack of good supplies for sample collection. To resolve this third issue, we then started distributing collection kits with the right tubes, the right media, swabs, and illustrated Bangla guidelines to improve overall sample quality. Soon, We were overwhelmed with the number of samples that poured into our lab. But we wanted to do more. Vaccines were starting to get into clinical trials, and in order to determine if vaccines in development in the developed world would also work in Bangladesh, we needed to know the sequence of the viruses circulating here. Remember that sequencing facility we built for meningitis? We started using that same facility for sequencing SARS-CoV-2. We were testing by the day, preparing for sequencing by the night. On May 11th, we became the first group in Bangladesh to sequence SARS-CoV-2. And what followed is phenomenal. This one action became the catalyst of unprecedented changes. Fellow scientists, journalists, reporters, my family, all over the moon, knowing that Bangladeshi scientists sequence the virus in Bangladesh. Often, we have to send samples to rich or developed countries to help us sequence and analyze the data. But this was all done by Bangladeshis. We were all over the news. My 88-year-old grandmother, my mother-in-law, all learned about RNA, viruses, and sequencing from my TV interviews. And even more importantly, within days, other groups joined us. Protocols started being shared, conversations erupted, passion ignited, and at least 6 other groups started sequencing. All because someone showed we can do it. Bangladesh has now sequenced over 300 SARS-CoV-2. In a country where research is the fallback option when you can't get into med school or engineering school, I have been getting calls and messages from young girls telling me they want to be researchers. They want to learn sequencing and bioinformatics and data analysis. Mothers have been writing to me saying they want their daughters to grow up to be scientists. My heart is more full than our sequencing machine these days. I hope as a nation we can sustain this momentum, this passion, the lessons we're learning for COVID 19 to fight the other diseases that plague our communities, like typhoid, dengue, or MDR TB. And if my team and I can indeed play a role in this change, I think my parents might just forego those application fees. Thank you.